Hello. Today's topic is affinity chromatography. Introduction to the technique we have to discuss in today's lecture. Right, when we talk about chromatographic procedures, we have various physicochemical differences of the molecules in a mixture that we are going to explore or utilize, make use of, and so that eventually we get the separation. Now, here in affinity chromatography, we have a specific feature of biological entities, that is the affinity of biomolecules for a specific and other another molecule. So wherever there is affinity for a specific molecule, these molecules having affinity for each other will interact through non-covalent binding, right? So the capacity of the biomolecules for specific non-covalent binding, that is reversible binding, we have to learn and use it in the affinity chromatography. Now, when I typed the word that is German scientist Emil Starkenstein, I got so many photographs on the Google, right? What I had typed in 1910, the German scientist Emil Starkenstein. When I type these words, so many different photographs they appear on the screen, and I have to choose the who is the right person. I'm sorry. Now the concern is, how do I choose this? So when I say this, I get so many photographs. So to choose from, I have to give some specific command selectively that will identify Emil Stark-Einstein, who has contributed to the development of affinity chromatography. So those selected keywords, when I put, I get the right photograph out of all these more than 20 photographs that have appeared. I have just shared a part of it, what had appeared on the Google screen. So what happened? You have given a selective keyword that would identify the right person. So selective keyword is going to be the ligand in the affinity chromatography, and the analyte will be selected using that ligand. So when I talk about affinity chromatography, in simple words to say, all of us we know lock and key model of enzyme and substrate interaction, right? Or how the protein interacts with its substrate. So there is biospecificity, right? Of course, it is not limited only to the enzymes. You can have given hormones that will bind only to a specific glycoprotein, that is the receptor for it, which is present on the plasma membrane. So antigen antibody is another example of biological specificity and the interaction which is going to occur through that. So suppose that an enzyme is to be purified from a mixture of different proteins. So as you are seeing in the diagram, you see three different columns. And in the column 1, you see column with substance S covalently bonded to the support material. The sample containing mixture of proteins, right? And the mixture of protein is labeled as P1, P2, P3, etc. Okay. Now these proteins are to be purified from the mixture, and when I say mixture, it is going to be many hundreds or thousands of proteins if I talk about cell lysate, for example. So from that I have to separate the protein of interest. Now the substance analog, for example, in enzymology you have heard the terms substance analog, inhibitor, activator, substrate, etc. So here I am going to talk about the word substrate analog, or the that is used for purification of protein, the the protein which is enzymatic in nature. Now what is analog? It is the molecule which resembles to the substrate, but it is not able to take part in the reaction. It is not capable of reaction. So such substrate analog, which is specific for the enzyme, is to be coupled to the column matrix. For example, in this support matrix, I am going to add a substrate analog so that the matrix is coated with analogs, and when I add protein mixtures, only the protein which is going to find this substrate analog as similar to its own substrate will bind to it. But because it is substrate analog, the protein will not interact with it, the enzyme will not interact with it, but just will have binding specificity. So reaction will not proceed, but it will remain anchored onto the column, and rest all other proteins will come down along with the mobile phase, right? So all other molecules which have no specificity for that ligand, what is the ligand here? It is the substrate analog. They will pass down the column, right? So in the second column you are seeing that is the P1 that is attached, but P2 and P3 and so many up to PS 10, 20, whatever there in the mixture, which do not have specificity for the ligand, they have come down in the elution fraction of the chromatography, right? So these are going to pass down and are collected separately, but the one which has affinity will remain in the column. After all the undesired components are removed from the column, now what the column is going to have is only the protein of your interest. For example, in this case P1, but your protein of interest is attached to the column, and it is in the is attached to the column matrix, and it is inside the column. Now you need to remove it from the column so that you can utilize it for your further studies. That is referred to as elution of the column. So when you have to talk about elution of the column, this protein enzyme has to dissociate from the ligand, right? So without undergoing any change, any structural or functional alteration, or disintegration or degradation, it has to get eluted. So column number 3, you have bound protein onto the ligand, and that is to be removed. Now here when you talk about removal of this bound protein or enzyme from the column, again you can take help of various basic principles, like maybe you are going to alter the pH to such an extent that the interaction between ligand and the protein enzyme will get weakened and protein will elute out in that mobile phase. So that is one way of altering the physicochemical property of the mobile phase, so to elute the column. The second would be key, you have to explore the biological affinity, and here mm, you can add an competitor, right, for the ligand. So the enzyme is going to have affinity for the ligand, that is the substrate analog, as well as the agent that you have added in the mobile phase. So if the affinity is selective for that protein, then that mobile phase will try to pull it into the mobile phase away from the column, and then it will run down the column and you can get the fraction collected. So to say, 8 minutes, someone is there. Yeah, to say this is overall schematic diagram to represent affinity chromatography working. Now let's go to the details of it. When I say affinity chromatography, it is a type of liquid chromatography in which biologically related agents are used to prepare the column, to prepare the stationary phase of the column, so that the analyte of your interest can be purified. Affinity, when we talk about, of course, the word describes the ability to bind selectively, and very importantly, it is a reversible interaction, because if your analyte of interest gets covalently bonded to the support matrix, you cannot ever have it, and you cannot do any studies on it. It is blocked forever. That should not happen. So it should be a reversible interaction. So, for example, the binding of a hormone to a receptor or an antigen antibody interaction. These are the few of the examples where bio specificity is availed or utilized. Right? Now, when we talk about the stationary phase of the affinity column, generally what we talked with respect to column chromatography in previous lecture, the same concept is here, that stationary phases you are going to have which are going to contain the solid support, so which is made up of agarose beads or silica particles. We have learned different types of support matrices. Right, out of this, agarose bead is one of the examples, of silica is another example that we have learned to details. So that could form the solid support. And one of a pair of interacting species is first immobilized. Means the antigen and antibody if you have. So for example, antigen is the protein of your interest. So antibody which is selective for that protein is to be, of course, the antigenic marker of that protein is to be prepared and is to be attached to your support matrix. The word is used is immobilization. So one of the interacting species is first immobilized to the solid support. And the target compound for the affinity ligand, now this what is immobilized on the column is the ligand, and target is your study protein, the protein of your interest. Then it is allowed to uh, run down the column, pass down the column in presence of a specific mobile phase, and then selective immobilization of the analyte onto the ligand occurs. Okay, now let's go to the point. Then why should we go for affinity chromatography when we have different methods for separation and purification available? Why to go into the by a principally very sensitive system, that is biological specificity? You are aware about antigen antibody specificity, how crucial it is when antigen it is to be tapped, a virus is to be purified, or as to be attacked. Body has specific again, like lock and key mechanism, selective functional groups are going to interact. And so these kind of interactions are very sensitive to ionic environment, the pH, etc. So why to go for it? The concern is affinity chromatography is highly selective in its nature, right? The interaction is very specific. And further, because it is being specific, it is very fast, you can say. It is going to be completed in a single step recovery of highly purified protein you can have. Now, another thing is very important is the potential of purification. So when you are going to talk about purification potential, when the selectivity filter is applied, of course, the potential of purification is going to be high. And how high? So it is going to be several hundreds to thousand times better purification or better yield you can get when you are going with affinity chromatography. The ability to concentrate the analyte, it is very important. So if you have large volume, so for example an enzyme is produced extracellularly by a bacterial cell, and your fermentation is done, and you have to purify the protein of your interest. So initial cell separation and all these techniques you would do, but total amount of enzyme in the initial phases of fermentation may be low, and you want to just check the check whether the enzyme of your interest is present in or not, but the volume is too high and the concentration is going to be too low, or maybe certain electrolytes in your body fluids, they are in um, very low amount, right, millimole quantities. So when you want to analyze such kind of things, or maybe a particular kind of mRNAs to be purified from total pool of RNAs, how will you go? So volume is high and analyte concentration is low. You have to concentrate that analyte so that further you can utilize it for assays. So as taking the example of enzyme produced by bacterial system, that initial fermentation, day 1, day 2, day 3, you take the sample out and you want to check for the presence of enzyme. Now initially it is going to be too low, but how much it is, and what is its activity, that all is to be characterized, right? So that concentration of the substance can be done using affinity chromatography. Other chromatography techniques would separate it out, but it will not be that selective as much as affinity chromatography is. For example, gel exclusion chromatography will separate it on the basis of molecular size, but related size proteins or related size enzymes will come together. You cannot you cannot exclude the one of your interest from the others, right? So in such cases, affinity chromatography is going to be very important, which is going to concentrate the substance from large volumes. the ability to separate proteins based on their biological function, where the active form can be separated from the inactive form, right? So you are aware that various enzymes and proteins, they are present in various structural forms, and the structural form will decide whether it is going to be functioning or non-functioning, and of course it is regulated by the microenvironment. But when you want to purify or differentiate active from the inactive one, different biological forms, if you want to separate, you can take help of affinity chromatography. Further, when you have to use such specific system of affinity chromatography, it can be like not only for enzyme assays or purification of protein of your interest, but further it has been of great help in biological scenario with reference to research and analysis. For example, it has been explored in many of the omics technologies. You have heard the word omics, like genomics, proteomics, metabolomics. Related studies, you need to have the particular interaction between two different proteins, or if you want to see is there any post translational modification happening after the protein is synthesized. You have to have a purified or separated protein from the bulk. So in such cases, affinity chromatography works really well. Now going to the next step in the discussion about how it really works. So I would say in simple words, it is capture of the analyte, then washing of the column to remove unwanted components from the mixture, analyte mixture, and then elution of the desired analyte. These are the three phases. To schematically show, here you are going to apply your sample to the column first, then you are going to allow the mobile phase to run down the column, and then you are going to elute the column, right? So this is your matrix, right? Support matrix, which is inert in nature, onto which the ligand is attached. So here, as you see in the schematic diagram, it is the antibody structure. So antibodies are attached here, and this is the mixture of analytes. So when it is applied, you see the specific analyte, specific protein binds with the antibody, which is the ligand, and rest all others which are not specific, they will not bind. So when you wash the column, step 2, wash the column, all of them will get removed from the column. And third step is bound analyte of your interest is to be now eluted. So for elution, you are going to have specific elution buffer, which is going to selectively elute the molecule of your interest without affecting its functionality and structural integrity. Right? So affinity chromatography is going to give you a very highly selective method specific for the specific analyte. The resolution is high, and capacity of protein purification is really very good. So we can uh, talk affinity chromatography with respect to research like protein protein interactions or post translational modifications to applications in therapeutics. That all we have to learn in consequent lectures. Now, here is the scientist Emil Stark Einstein, which I had referred in the very first slide about. So, this person in 1910 just put the concept that we can resolve the macromolecular complexes via their selective interactions, provided they are immobilized. So, they are immobilized onto some chromatography substrate. Of course, this concept was first applied with respect to biological samples, that was alpha amylase from the liver, and it was purified. Of course, the term affinity chromatography wasn't coined by them, it was introduced later, almost after 50 60 years, that is in 1968. It was introduced as affinity chromatography term. In between, lot of research has taken place and the technique had advanced. For example, the technique of enzyme purification via immobilized substrates and using inhibitors. So, substrate analog and inhibitors, you know these terms with reference to enzyme. So, this was explored. Then, the activation of the matrix, right? So activation of the matrix is required. What is it, how it is done, that we will learn in the next lectures. But activation of the matrix so that it is active and binds selectively with the ligand, that is very important. And then after it is bound with the ligand, ready to interact, again it needs activation, which we have learnt in column chromatography. So activation of the matrix, that was done in 1967. You have the references talking about it. And then another concept that is very important that we have to learn in today's lecture is the spacer arm. So spacer arm is added to the system so that efficiency is much better and resolution is really good. So that was done as early as in 1968. The reports are there. So this is in short the basic history, but why I talked about it is these are the important concepts. So first is interaction between the uh, macromolecular complexes. That is the first keyword. Second keyword is immobilization of one of these uh, uh, pairs of molecules which are interacting. That is second keyword. Third is use of substrate analog or inhibitor as an example of one of the interacting molecule that you are going to choose to separate the molecule of your interest. Fourth is activation of the column and matrix, and fifth is spacer arm. These are the important key concepts when we talk about affinity chromatography. So I had referred to these. Now look into the important steps. Three of these I'm mentioning here. The affinity matrix, right? Now the matrix is not simple as column chromatography that we learned in simple liquid chromatography. That is, you have only a silica gel or agarose or dextran or Cifadex, which is the brand name of it, the are there, but you have specialized matrix, so affinity matrix, on which you have immobilized the ligand, right? Now, how it should be? It should be macroporous with high chemical and physical stability. That is there in silica gel and all other matrices that are commonly used, and should be the one which will selectively capture or separate out the target molecule of your interest. At the same time, the phenomena that we had talked in the previous lecture, that is, it should not uh, compromise the efficiency of the column. That is, when you are filling the column and passing down the mobile phase down the column, because of the pressure generated of the material inside the column itself, its own weight is going to create pressure. And if that happens, then the lower layers in the column are going to get compressed and efficiency of separation will be hampered. So you have to take care that these are having a good separation, and it is not going to get compromised when the column is going to actually run, right? It's not that always HPLC is run. Without high pressure application also, or MP, that is moderate pressure liquid chromatography, MPLC or MPCC, you have to run liquid chromatography at normal gravity force. They will be running on their own, right? So you have to take the one which is not going to have any specific adsorption. So it should have low non-specific adsorptions, and it should maintain a good flow rate. That is what I was talking about. Compression if occurs, then flow rate of the mobile phase will get affected. So it should retain its good flow rate, and for that purpose the strength of the gel beads of the matrices is very crucial, so that you can get a high throughput processing, right? Of course, it should be comparatively inexpensive, readily and easily available, and it should be simple, because when it comes to uh, research or industrial scale, all these things are going to matter a lot. Now, second thing comes is selection of the appropriate ligand that you have to add to the particular affinity matrix. Now, add to the support and develop the affinity matrix. So ligand is to be chosen. Now, to know, you have to have a basic knowledge in biology as well as chemistry, because the biological materials which are going to show selective interactions are to be chosen, right? Biotin evidence system, enzyme substrate system, these are the things that we are talking about. But how do they interact with each other? Which kind of forces are taking part in? Which kind of charge or what kind of bond is getting formed between them? That is to be understood. We should, for example, if you take example of substrate and enzyme, then you are aware about what is the catalytic site, right? Which is the functional site, which is the support site, that is which is going to arrange the enzyme molecule in its functional uh, orientation or form, and the functional groups in the catalytic site which are actually going to react with the substrate. All these things are to be known. Otherwise, when you are putting this ligand onto the support matrix, the functional groups or the important charges, if they get locked, that is, they are going to interact with the support matrix, agarose, dextran, silica gel, whatever matrix you have chosen, if they interact with it and get blocked, the entire functionality may get compromised. So you have to have basic knowledge of the uh, interactions between the ligand and the target molecule, so that the ligand must specifically bind with the target molecule. If these active sites of that ligand or functional sites or the functional groups of the ligand, they interact with the support matrix, they will not remain open for the target molecule to attach with, right? And for this you need to know basic chemistry. And of course, another thing is, it's not only support and binding of the ligand onto it and then binding of the analyte onto it, but elution also. So whatever kind of elution buffers, the ionic environment that you are going to provide, that time also it should remain stable. If the ligand undergoes changes and comes out of the support matrix, again it will be a challenge to purify your analyte, right? So it should be stable under elution conditions also. So whether the ligand should be interacting with the analyte by covalent bonds, no, no, it should interact with the analyte by specific but reversible attachments. But when it comes to binding of ligand onto support matrix, column matrix, that time it should be a covalent attachment. Right? Now let's look into schematic representation of the same concept. That is, when I talk about the important steps in the process, first is the resin matrix, first component, then the linkage, right, of the resin matrix, support matrix, column matrix, whatever you call it, with that of the ligand, and then addition of your sample. But again, here another term has got introduced, that is, it is not uh, the addition of ligand directly, but a spacer is to be added. So spacer arm is a concept that needs a little highlight. Can you observe this diagram? This is the support matrix, right? Now onto which you have something like a stand or a hook, right? So the arm of the hook is the spacer arm and the hook is the ligand. That is how schematically you can show the diagram. So this is the mixture of your analytes, which are structurally similar, functionally also may be similar but not same, needs to be purified. You are going to use a specific ligand, selectivity is there, so analyte will bind onto it and all others are going to run down. But schematically, to understand the concept of spacer arm, you can just imagine, instead of this horizontal line, if this ball, this is horizontal line and ball, so ball is your ligand, the horizontal line is your spacer arm. If this spacer arm is absent and this ball, that is the ligand, is directly attached to your support matrix, what will happen, right? So that is a concern that is to be talked about. Now, if I go to the next slide, when we will see the effect of addition of spacer arms, right? Spacer arm, like for example, if you have a single stand a single vertical rod, and onto which you have to keep your say raincoats or bags. How many you can really anchor on it, just on the tip of it? But it has got side hooks, okay, maybe more than that. But if the side hooks are closely spaced, then really all these bags will be like pushing each other for the space. There will be not enough space to hang more than two three bags, right? But if I take horizontal hands away from it, so the stand is having the vertical rod, and from the vertical rods you are going to have the horizontal rods extending, which are going to act as hand with a hook. So you can attach multiple things onto it, and they will remain stable also till they are removed, right? So when I'm talking about this spacer arm, why it is required to have a spacer arm? Very importantly, as I said, to hook your analyte selectively onto the ligand, it is very required. Otherwise, steric hindrances, right? So steric hindrances, for example, as I said, on a vertical rod, how many bags you can hang on it, right? Two, three, four, one above the other. What after that? The rod will be surrounded by so many bags, and the bags will fall away. Better if you have horizontal rods attached to it, you can very well hang multiple bags onto that. So that is what is the importance of having a spacer arm. So the ligand is, if directly attached to the activated group of the support, the micromolecules, that is your analyte, will directly anchor onto it, and then they may have a problem like steric restrictions. So when I say steric restriction or steric hindrance, that means there won't be any proper adsorption occurring onto the matrix. What do you want? You want your analyte of interest to get hooked onto the ligand, right? But if they start pushing each other because their interactions with the ligand and the interaction of the ligand with the support matrix, if crowding is there, then steric hindrances are going to uh, not give you good adsorption to the matrix. So to remove this difficulty, it is required that you add a spacer. The name itself is saying to create a space. Spacer is added. So spacer is added between the activated group of support and the ligand. So spacer, the word is used as arm, the spacer arm, right? So due to this, the ligand is going to be like something away from the support matrix. It is like getting projected at a particular distance from the matrix, so that when a desired analyte molecule, which is having selective specificity for your ligand, will bind to it, but it will not have any steric hindrances. Of course, the spacer arms, there are variety of them are available. For example, you can use diamino dipropylamine. So diamino dipropylamine, you get to know the nature of the molecule, right? That amine group is going to be the functional group, and rest is your diamino is another one. So diamino will react with support matrix, and amine may be available for your target. And in between you have dipropyl. So when I said dipropyl, it is to hydrocarbon, right? So you are going to have such spacer arms, so in which you have examples like aminohexanoic acid or diamino dipropylamine, diamino dipropylamine, right? Aminohexanoic acid, or I can say 1,4-bis epoxy propoxy butane, right? Epoxy propoxy butane. These are again epoxy propoxy, so these are going to be the particular groups which are going to interact, one with the support, another with the ligand, and in between you have a hydrocarbon chain, right? So like that, hydrophobicity, hydrophilicity, functional groups, polarity, all these things are to be considered when we are talking about the spacer arm, right? Now let's go to the next slide. When I say the column matrix now, so in the matrix, support matrix, what you have? Silica gel, you have agarose, dextran, uh, n number of possibilities of matrices that are commercially available today. Even polymers, right? Natural polymers, so synthetic polymers also are available. We have talked about it in column chromatography. So they are available once that these column matrices are activated by addition of the ligand, right? And then you are going to have the affinity column. Now, what are the different types of supports? So many are there, as I said, but they can be classified as porous ones. So silica gel or cellulose or cephadics, dextran, agarose, all these are porous things, right? Macroporous. And we have seen that they are available in different sizes also. Particle size and pore size also has to be very specific when you are preparing a chromatographic column. And based on that, the readily available columns are there in the market also. You have to specify what is the particle size, what is the pore size, diameter size of the column when we are talking about the specific support matrix. So you have to say, I want Cephatics column of